So hi everyone, I'm Roey. I'm also from Aviv Mezer's lab here in ESF, like Shai. And I will tell you how we combine different types of MRI scans in order to better map white matter tracts in the human brain. So the white matter tracts that we're talking about, they take the result of local computation in one part of the brain, and then they communicate it to distant parts of the brain. And to study them in vivo, what we do is we use a method called diffusion MRI tractography that we just heard about as well from Roey. And we try to use it in order to map and reconstruct the structure of these different tracts. Now, it has many different uses. One of them is such a connectivity analysis where we look at the brain as a network. Another form of this is the human connectome. So the connectome is defined as this network or matrix of connections between different brain regions. And we think about it as, let's say, the infrastructure for cortical communication and information processing. So in some sense, it should tell us something about how brain systems are organized or how they change upon injury or disease and so on. But we have to remember that these edges or these connections are not just abstract edges on a graph or in a matrix, they're actual physical entities that we can study. And indeed, many papers have shown that the physical properties that you can measure of these tracts correlate well with different cognitive abilities like reading and vision and so on. So in this sense, being able to accurately map these connections is really crucial for studying the function structure relationship in the white matter in the human brain. So to do this with tractography, the idea is that the diffusion MRI signal is really sensitive to the displacement of water molecules. And when water molecules are restricted by their environment, this displacement over time reflects the local geometry of the tissue. So in a, let's say, a very general kind of diffusion MRI study, what we do is we take a series of diffusion-weighted images, and we fit some local model, let's say the diffusion tensor model, and then we try to follow the most probable path to reconstruct the streamlines that represent the geometry of these fibers. Then we can start studying the connectome and so on. So this sounds great, but in fact it has some very major limitations that we're trying to address in the lab. So one of them is that I told you it tells us something about the geometry of the tissue, but it doesn't tell us the whole story. And in many cases there are fibers crossing each other in the brain or kissing each other. And here you can see an example of a voxel where we have two subpopulations of fibers, each going in a different direction. And the signal that we get is some weighted average of these two subpopulations. When we fit a model to this, the model doesn't really capture the underlying geometry. And this is not just a local effect, it soon propagates to create many global effects. So if we take this toy example here and give it to a tractography algorithm, it cannot differentiate two very different scenarios. So even if the true fibers are crossing each other, it might also create fibers that kiss each other and then diverge again. So we want to study the connectome, we want to understand how it changes in different states, but we have two very, I would say, basic limitations. One is we're not even sure which connections exist and which don't. We want to be able to eliminate false positive results. And the other is, even if we do get all the geometry right, how do we group streamlines together? How do we say where one system begins and the other ends if we want to study specific tracts? So the question we asked in the lab is, maybe we can take some complementary information from other kinds of quantitative MRI, not just diffusion, and then integrate this new information to try and tackle these current challenges of tractography. So we'll talk very briefly about how we address the first challenge, just one slide, and then I'll talk in more detail about how we try to tackle the second challenge of identifying specific tracts. So as for this first challenge, we focused on the optic radiation. So you can see it over there in a dissected brain. You can see it from below. The optic radiation connects the thalamus to area V1, the primary visual cortex. And we know that it's really hard to accurately find in tractography, just in naive ways of doing it in tractography studies. But what we do know is from post-mortem myelin staining studies, the optic radiation is really shows a very distinct signature of high myelination. Luckily for us, in vivo this is reflected by T1 max. So T1, the longitudinal relaxation time, is very sensitive to the myelin content, among some other stuff. And you can see it here that the optic radiation really stands out as a very dark stripe of low T1 values since it has high myelin. So what we did in this work, we took a very naive approach for finding the optic radiation. Just take all the streamlines between the thalamus and area V1. You can see it's a very noisy representation. It has many false positives. And if we just take the T1 map and integrate some information into these or onto these streamlines, we can use what we call T1 filtering to get rid of these and get a much cleaner representation that actually corresponds well with what we know about the anatomy of this structure. So this was how we tackled the first challenge, and this gave us the feeling that maybe we can try and tackle the other one. Maybe there is some difference between different tracks in terms of their signatures in T1, and it could help us make the distinction between different tracks. So here we focused on another visual tract. This is the VOF, the vertical occipital fasciculus. And as you can see here, it connects the dorsal and ventral visual streams. It's really interesting. It draws a lot of interest lately, mostly because it is thought to be what allows us to integrate information between these two very different streams. And one thing to know about it, already known in 1909, is that the VOF is pretty hard to delineate, especially around the anterior part, because there's another white matter tract there, the posterior arcuate fasciculus, probably not a classic visual system, and they're so close together that they often form a continuous sheet of fibers, and it's hard to say where one of them ends and the other begins. And this is of course also true when we go to in vivo studies, when we try to find, let's say, all the vertical streamlines in the posterior part of the brain. It's one big mess. You can't really say where one of them ends and the other starts. So we thought, okay, maybe their geometry is very similar, but probably or hopefully their microstructural signatures might differ, and we might be able to use this. So here you can see an Excel slice of color-coded diffusion data. So blue means it's vertical, and you can see both the posterior arcuate and the VOF here. If we just go to a T1 map, you can see that these two clusters correspond to very different T1 values. So the T1 in the more anterior cluster is higher, and in the more posterior cluster it's lower. It has lower T1. So the question we asked is, can we find this effect also at the streamline level, not just one slice? So if we go to the streamline level, we can take our streamlines and overlay them on top of a T1 map. And then what we do is we take, for each streamline, we can calculate some characteristic T1, let's say the T1 median, that's what we did. And if we color these tracts by the T1 median, you can see that there are very distinct clusters. Each one of them has a distinct and unique T1 signature. And hopefully we can use this signature to delineate the VOF. So I'll just briefly say that there are methods to find the VOF. They're based on spatial criteria. So we're trying to find ways to use microstructure instead of heuristic macrostructure. But using these methods, we were able to take the published methods and show that across our 89 subjects, indeed, VOF has lower T1 compared to posterior arcuate. So now, just a question that we ask is, can we use this information to actually define the VOF, to separate it from the posterior arcuate? So, yes, we can. Thanks. So now we can take each streamline and represent it in some two-dimensional space. Each streamline is now one point, and you can see it's T1 median against its location along the anterior-posterior axis. And we're just looking for the point of sharpest increase in T1. So it's a very simple but robust algorithm. And when we put this border over there, what we get is what we call VOF T1. It's VOF defined by its T1 signature. So we repeated this for the entire almost 90 subjects that we had, and we saw a very similar trend across all subjects. And then using this identified VOF, we could start asking things about the tract itself, or what can we learn about it now that we identified it and we're pretty sure about its identification. So one thing we did here is just one example. We took a surface-based approach for studying the cortical terminations of the VOF. So this is an inflated cortex, and you can see it in template space. There's a heat map that tells us for each point how many subjects had VOF ending in this location. So you can see, first of all, that it does project where we expect, ventral and dorsal visual streams, and also that it's very consistent across subjects. What you can also see in blue is all the fibers that we threw away, the anterior ones that we eliminated. And you can see first of all, most of them actually correspond to what we know about the posterior arcuate, which is interesting. But moreover, they're not classical visual regions. Moreover, if we look at this heat map and compare it to known atlases, you can see both in the dorsal views on the sides, where we have a functional atlas of the dorsal visual stream, and here in the ventral part where we have a cycloarchitectonic atlas. You can see that overall these cortical projections really are restricted to very specific regions, which pretty much agrees with what we know about the VOF from the literature. So overall, we can conclude that T1 can be used to delineate specific wide matter tracts based on their unique microstructural signatures. I'll just tell you very briefly that we have a poster about another tract that we tried this approach on. It's the superior longitudinal fasciculus, the SLF. We know that it's actually made of 3 Griffin tracts. And again, we found that it has a very unique signature between SLF3 and SLF2. We used a very similar approach to just find the border between the two, and when we compare it to another published method for finding the SLF number 3, we can see that we get very similar results in these subjects, but also, of course, in many other subjects. So just to summarize everything, we saw that diffusion MRI is inherently limited due to some local ambiguity in how we can represent the geometry of the tissue. But then we can take complementary information from relaxometry MRI, like T1, and we find that it does hold valuable information for identifying specific tracts, both in the case of the VOF, but probably in other parts of the brain. We now know something about the SLF, but maybe in other parts as well. So with that, I'd like to thank all my lab members, and especially Aviv, the mentor, and Dean, who took part in the SLF project, and of course you for listening. Thanks.